Hello everyone and welcome to today's webinar, Immunophenotyping Unleashed, a real partner in panel design. My name is Phil Donaldson and I am the Market Development Manager for Flow Cytometry for Thermo Fisher Scientific in the Asia Pacific region, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more, visit www.thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We have Takanori-san from Japan and Sukyong from Korea, our technical experts on standby too. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. With that, I'd like to now welcome our speaker, Dr. Patricia Haus, Supervisor for Technical Application Support based in Darmstadt, Germany. For a complete biography on our speaker, please see the Presenter tab at the bottom right-hand side of your screen. Dr. Haus, welcome. You may now begin your presentation.
Hello. Thank you for inviting me today to speak about our new flow panel design service from Thermo Fisher Scientific. My name is Patricia Haus, and I'm located in Europe, to be precise, in the Midwest area of Germany. I'm the supervisor of our new flow panel design team. I started my journey as a technical application scientist here with Thermo Fisher Scientific already 8 years ago. And I was supporting my customers with cell biology, but also cell analysis questions before I became now the supervisor of this team. I have a PhD in biology with a focus on drug development, and I always worked before I joined Thermo Fisher Scientific in cancer research. I'm presenting today from our Darmstadt facility. Even as you cannot see, let me invite you whenever you are around here in Germany and you want to learn more about flow cytometry, please let us know and we will be more than happy to arrange a demo or even a training. But let's get started with our agenda for today. So we will start with a very brief introduction to multicolor flow cytometry. I know that every one of you is bringing already a huge background about flow cytometry and you have all of this knowledge. But let me bring us all on the same page first, speaking quickly about multicolor flow cytometry, but also then about what flow panel design actually is. You will hear then a lot more about what our flow panel design service can offer to you, who we are and what we offer basically. And afterwards, my team will provide you with some tips, but also some tricks about successful flow panel design. And finally, we will have more than enough time to go through all of your questions. But if you have now already any questions, please feel free to use our chat. My team members Saki Jung and Takanori will be happy to answer all of them. But as I said, we will have enough time at the end that you can unmute yourself and we will go through your Q&A session. My apologies, and that's the last word before we are actually starting with my presentation. My apologies that this is a pre-recorded video. Based on the time differences, we believe that this is the best option. So let's get started with the basics of multicolor flow cytometry. And I said it, I will make it brief. As you might, or as you for sure will know, flow cytometry is our choice of technique if we want to have the flexibility, but also the sensitivity to detect fluorescent signals on a single cell level. This allows you to analyze multi-cell populations and quantify expression levels in one sample, but on a single cell level. A flow cytometer allows you to distinguish cell based on size, but also cells based on their granularity. you know, this is where flow cytometry came from. That was the basics. But nowadays, we want to know so much more, isn't it? And together with fluorescent labeled antibodies, you can detect cell surface molecules, but also intracellular molecules. Within the instrument, the cell passes the laser, one cell at a time. This is why it's ensuring that what you're seeing, what you're detecting is really based on a single cell event. So flow cytometry generates all of those data super fast. And that is usually why you would choose this technique over other techniques like a confocal microscope if you want to process a lot of samples or if you want to know a lot about one cell. So moreover, what you can do is you can detect several targets in one sample, showing you a more complex view of a situation. As an example, if you're treating your cells, let's say with a new drug candidate, you might want to know how this drug affects your cell, not only on a live-dead scenario, but also on different specific targets within or outside the cell. Or you want to process so many more samples, isn't it? So let's say you have a huge campaign with hundreds of patient samples and you want to detect within all of those patient samples the status of a certain target. Then this is also usually the technique what you would choose. However, some of the challenges you might face are that you require to have a very good panel design, and that's when we come into the picture, or you have to choose your controls very wisely. So let's have an open discussion about that as well. Or you have to establish your protocol properly, and just saying it one more time, this is also something where we want to help you. If you want to know more about flow cytometry, please find below one of the links to our web pages, which gives a very brief introduction, but a little bit more detailed than what I was doing today into flow cytometry. So, but what is flow panel design? Flow panel design is choosing the right antibodies with the right fluorophores for your experiment. Basically, what you want to ensure is what you are detecting is actually answering your question, isn't it? So you want to see all of the populations well and distinguished, and you want to quantify the expression levels. One of the biggest challenges in multi-parameter flow cytometry is selecting actually the right combination of fluorophores and the antibody conjugates. But only this makes it possible that the need for compensation, but also the spillover adjustment is kept to a minimum. So by choosing the products and the conjugates very wisely, you can ensure a very high standard of your quality, but also you can ensure that your data set is accurate. And at the end of the day, it is super important that both combinations, so for which target do we want to use which antibody conjugate, that this is under the optimal circumstances. So again, if you want to learn more about panel design, I provide 2 links to very nice overview pages. There is a lot what you can learn about panel design. You will learn a little bit about it today. If you want to continue learn more about it, please find the links below, but you can also contact us. So, but let's hear a little bit more from Berencia. Berencia is one of my team members and she's located in the US. And she had the chance to work with our marketing team together to speak about what she finds most interesting about her new job, but also about this panel design service itself. So let's hear from her what she finds exciting about that.
I've always been pretty curious as a child. My mom, she's a physician. I always liked when she talked about the various cases that she'd have at the hospital and how to come to a diagnosis. My name is Patricia and I'm a technical application scientist at Thermo Fisher Scientific. I work on a team that provides flow cytometry panel design services. My first job in a laboratory, I was doing various bacterial genetics techniques. I really appreciated my PI, my mentor, the senior member in the lab who helped me understand what those experiments were. That was my favorite part about the lab. It's very community-like and it kind of an art to it. I love that element of science. It's not just extremely structured, it's artistic in a way. What really inspires me to get up every day and start my work is I love getting a new panel request come to my inbox. It's like having a new puzzle that you need to solve every day. One of the challenges that users face, even if they have a well-designed panel, is understanding the importance of good quality controls. Although your panel may be well-designed in theory, with the correct fluorochromes, the correct markers that you're interested in, a lot of the prep work that goes into running your sample will dictate whether the panel actually performs well. Sometimes when you're really into your research, it can be really difficult to try and learn a brand new topic. That's where I come in to help with panel design, already having this knowledge available so the researcher can focus on their sample prep, their question that they're trying to answer, and all the other experiments that may be involved with their question, because it might not just be flow cytometry. This panel design service provides that resource to researchers who really need that dedicated time and attention. All of us on the team are a global network of scientists who are fully dedicated to panel design consultation. Each of us have a unique perspective, and this allows us to leverage a wealth of information that the researcher has access to when they decide to use our service. I genuinely want to support the researchers that are doing important work in their discipline. I want to see them use this technology to its fullest potential to be able to answer those difficult questions that lead to the most amazing discoveries.
So to give you a better idea of our work, I wanted to share two examples today. So those are two examples of the most common request types and how we do the panel design together with you. So one of the very common questions what we get is about human PBMCs, especially about PBMCs actually. So I want to detect CD3, CD4, CD7, CD56, and CD16 in my human PBMC sample. And I want to discriminate between live and dead cells. I have an Attune NXT instrument with a red, a blue, and a violet laser. Can you help me? So we already have very valuable information. Even as this is a very small email, we know most of what we actually want to know before we are starting with the panel design. And what I want to guide you through is now the 4 steps, what we are usually using to gain all the information to be able to provide you with the optimal panel design. So the first step is knowing your cytometer configuration, or that we know your cytometer configuration. In this case, we have an Attune instrument and we know the configuration. The next step after that is the antigen selection itself. We know already what the customer wants, and we know in which sample type he wants to detect that. But to do our job optimal, what we also would ask you is the why. So we would come back to this email and ask you, what is the aim of your experiment? Why do you want to detect those markers? After that, we would choose the optimal fluorophores in step 3. As an example, we would think about the expression levels and the brightness of the fluorochromes. But only by considering what you want to achieve, we can ensure that we are choosing the optimal fluorochromes for your targets, especially if it's getting a little bit more complicated, if we are using all the channels of your instrument. And then finally in step 4, we would provide a list of available products to perform your experiments. So another example, again, a very popular request. I designed a panel, but I'm not sure about the quality. Could you provide some feedback, please? So the customer sent us the data on the left side. And as you can see, the customer will have a very hard time resolving this double positive population with this combination of fluorophores. The reason is that we see a lot of spreading from the fluorophores on the x-axis into the adjacent detector on the y-axis. Only by choosing now a better fluorophore combination, the customer will be enabled to see all the populations well enough to actually get some data out, isn't it? So what we offer is now our expertise and our knowledge to turn a panel with improvement opportunities, as you can see on the left side, into a better panel, allowing you to detect all of your populations like it is on the right side. And by that, the customer is now able to detect his rare populations. Now, I want to introduce you to our flow panel design service with Thermo Fisher Scientific. Within the last years, flow experiments became more and more complex. That is the reason why we decided as a company that we want to invest in this area and offer our service. Back when I did flow experiments, when I started doing flow experiments, we were already happier and we believed that we got a lot of data out of an experiment when we had a viability dye, a marker for the population of interest, and a marker of a specific structure. But nowadays, the majority of our requests are actually for panels around 12 colors. So the customers want to know more and more about their experiment, about their cells, what is actually happening within the cell. What we are seeing is that there is actually a trend that it is becoming more and more complicated. It can easily go up to 20 colors. And if you're considering doing spectral flow, it can even go up to 40 colors. And all of that creates a completely new challenge because it requires so much knowledge and a very good understanding of the products which are on the market and which are available. And this is the reason why we believe that we want to make your life a little bit easier. And what we wanted to do is offer you a self-service, but also our expert support. Let's have a look into the self-service. What you can find here are 3 of the most popular tools if you want to design a panel. The first one is the antibody search tool, which allows you to search for products, especially antibodies, and makes it very easy to narrow your search down. On the other hand, we offer our panel builder tool. This allows you to see what your design looks like. And on the other hand, if you're, let's say, not 100% familiar with some of the dyes, then we offer our Spectra Viewer tool. All those tools will make it easier to see the properties of your conjugate, but also gives you easy access to the corresponding products. But in case you need our help, we are more than happy to help you. So what we are offering now within the Flow Panel Design Service is helping you to ensure that you have the right products for your experiment. So I added the link to our web form, but also our email address, flowpaneldesign@thermofisher.com. Do not hesitate to contact us with any question. And please, if you want to speak about your flow panel design, or if you want to discuss flow panel design in general, just give us an email, send us an email, or use the web form. And it's super easy. Just send us the email, but please consider, and I spoke about that before, include a couple of things. The first one is the instrument type and your configuration. Then the second one is tell us the why. So why are you planning, what are you planning to do, and why do you want to do that? And the third one is if you have antibodies already available in your lab and you want to use them, or you want us to consider using them, please send us a list of your available antibodies and we will take them into consideration. So let me explain now a little bit more in detail what our service is about. Let me start by saying that we are a completely free of charge service. You do not have to buy a product or pay a fee if you want to use our service. Everyone in my team is a flow expert and will design your panel together with you. At least 2 scientists will have a look over your panel to ensure that we have the best quality. But please consider that even with all of our knowledge, those are theoretically planned and designed panels, and they might require some improvements. So this is the reason why we don't stop after the panel design itself. What we offer additionally is that we are giving you, or we offering our help establishing your panels and even troubleshoot your experiment if the results might not be optimal from the beginning. So you can expect a first answer within 24 hours. And after we got all the necessary information, let me repeat it here one more time. Instrument configuration, what do you want to do, and what antibodies do you have already in the lab, and the antibodies what we should consider putting into our panel. So if you have all this necessary information, you will receive a first panel draft within 72 hours. In case we couldn't hold the 72 hours, let's say you have a very complicated multicolor panel, we would keep you informed. So we would tell you when can you expect our panel to be ready, our first draft to be sent out. After you confirm the panel, you will receive a product list from us. And what we would do as well is involve our sales colleagues, and they contact you with a quote then. So we are a global team. We have team members all over the world. As an example, we have team members in the US, we have in Europe team members, me in Germany, and we have other team members in Spain. Then we have in Asia team members in Korea, Japan, and in China. So our service is cross-regional and on a global level. So what does that mean? It means that it doesn't matter where you're from. You can write us from Spain, but you can also write us from China. It doesn't matter where you are. But it can also happen that you wouldn't get the answer then from your region. So let's say you're writing us from Korea after working hours, you might get a reply from our European team. And that ensures that you get the fastest answer, what we can provide at that point. So let me summarize the key reasons why you should choose Thermo Fisher Scientific and our service. Let us do the work. And that's basically saying all of it. We know that flow panel design can be difficult and very time consuming. So if you're just getting started with flow experiments and you need to learn more, please do not hesitate to contact us. We are more than happy to design together with you the panel and take you on the journey to learn more about panel design itself. Or if you're super experienced and you don't have the time, we would be also very happy to design a panel for you, work with you together, but taking a little bit out the panel design itself, giving you back time. So you don't have to be a panel design expert to get your experiments up and running, and you can spend your valuable time more on your other duties. What we will do is we will guide you through the process. And if you want to know more, we will be happy to share our knowledge. What we want to ensure is that you're doing the best experiment you can do under the optimal circumstances. And what we offer is scientist-to-scientist support. Thermo Fisher Scientific, on the other hand, offers the largest number of fluorophores, including unique brilliant dyes, but also our NovaFluor dyes. And let me emphasize one last point, because this is something what makes me very proud about this service. This is completely independent from your instrument type. So you can have an attune instrument, and I'm more than happy to discuss things about an attune instrument with you, or let's say, let my team discuss it with you together. But you can come with any other flow cytometer what you're using in your lab. And we are considering your conjugates. So if you have already 10 conjugates in your lab, antibody conjugates, then tell us what you have already, and what we will do is take that into consideration. But what we will also tell you is, this might not be the best panel then. So what we will tell you is, this is the optimal choice, and this is the second best choice. So in this case, you can decide what you actually want to do. So, but now enough about me and panel design service. I hope you got a nice introduction to what panel design service is and what it might offer as benefits for you. But I'm sure that you attended today also to learn more about panel design itself, isn't it? So what my team is sharing today are 6 of their tips and tricks, and Saki Jung, my team member from Korea, will start speaking about our spectra viewer and how it can help to design a flow panel.
When designing a flow panel, spillover is one of the most important factors to consider. To avoid severe spillover, you need to know the spectrum pattern of each fluorescence. Thermo Fisher Spectra Viewer can help you with that. As you can see on the screen, in the Spectra Viewer, you can input lasers, emission filters, and fluorophores. Selecting a fluorophore of interest displays its excitation and emission spectrum. And you can input multiple colors to see how much they overlap with each other. If lasers and bandpass filters are also set, you can check how much spillover occurs numerically. If you click the spillover table button at the top and designate a filter stop for each fluorescence, you can see the value of each fluorochrome detected in the designated filter. Check spectrum overlapping and minimize spillover with Spectra Viewer.
Hello, my name is Takanori Maeno. I'm Technical Application Specialist from Panel Design Team at Thermo Fisher Scientific Technical Support. Today, I talk about topic of fluorophore and stain index. The brightness of the fluorochromes is of course important for data resolution in flow cytometry, but it also depends on the difference between positive and negative populations. In flow cytometry, this brightness index is called stain index, which is a difference between the median brightness value, which is called the MFI value of the positive population, and the MFI value of the negative population, divided by twice the standard deviation of the negative population. The larger the stain index, the wider the separation between positive and negative population. For example, if the positive population is lower, or the variance of the negative population is wide, the value will be small. The signal-to-noise ratio often heard in fluorescence microscopy is the MFI value of positive population divided by the MFI value of negative population. Therefore, even if the distribution of the negative population is wide, the same equation is used even if the MFI value are the same. So, the stain index is a great indicator of fluorescence brightness, and it is correlated to how well your populations resolve when looking at your flow cytometry data. And each manufacturer may have its own stain index that is assigned to the fluorochromes in their catalog. They will often provide a list of fluorochromes indicating brightness in hierarchy. The stain index is an important factor when building multicolor panels, while also considering the expression level of the sample and fluorescence spillover.
Today, I'm going to walk you through the best controls that you need for your flow cytometry experiment. The first control that I think is really important is an unstained control. This control is your cells that are treated the same way as your sample, but with no antibodies added. The reason why you would want to use it is because some treatments affect the autofluorescence of your cells, and it will give you a baseline level readout of your cells on your cytometer to see where the natural fluorescence falls. The next control that I think is important is a compensation control. The compensation control is a single stain control, which can be beads or cells stained with only one of the colors, one tube or well per color. You need it in order to tell the instrument where exactly is the true fluorescence for your particular fluor. If you're assigning FITC or PE to a certain detector, there will be spillover. And in order to subtract that spillover, you have to provide the instrument a control to do that mathematical calculation. You need to make sure that you're matching the fluorochrome that you're using in your sample with your actual compensation control. And you also need to match the lot for any tandem dyes, which is like PerCP or PerCP Sci 5.5 or PE Sci 7. Make sure you match the same lot when making your single color control. The next control that I like to bring up is the isotype control. A lot of journals do not require it anymore, and that is because it is not as helpful as we once thought it was here in the flow cytometry community. I highly recommend that you research this topic yourself because there's a large conversation surrounding the isotype control. Essentially, the reason why it may not be the right control for your experiment is because it is really difficult to make a proper isotype control. You have to actually hit these 4 points here, make sure that it's the same species and IG isotype, which is fairly easy, but trying to match the fluorochrome to protein ratio, the concentration, the labeling conditions, that can get really difficult. When in fact, the next control that I like to mention is the FMO control, the fluorescence -1. This is sort of seen as the replacement for the isotype control because it actually truly does help you to set your gates properly. What it does is it accounts for spreading error, which is a phenomenon created by the flow cytometer, and it's a sort of a photon counting error that happens when the PMT detector is amplifying that signal. And so with the fluorescence -1, it's not necessary for every marker, but it's just important for the ones where you don't expect a distinct positive and negative population. This leads me to the biological control. And you can see here on this plot, this is the smear that I was referring to, where it would be kind of difficult to know where exactly your gate should go without the proper control. So with a biological control, it's important because you can basically use it to understand the difference between the base level of expression and any increase in expression due to upregulation, which may be caused by any of your stimulatory conditions. As you can see in this figure, the unstimulated control is actually the best option. So this is why I brought this here, because basically it's important to know that there are these different controls out here because one of them might be better than the other. So in summary, if you want to think about a control related to your setup of your flow cytometry experiment, your instrument, you want to think about your unstained control and your compensation control. For specificity or gating, you want to consider the fluorescence -1 or the isotype control. For biological comparison controls, you can go with a positive stimulated control or a negative unstimulated control. Thank you so much. We are available to you to help you build the panel that you need for your experiments.
Hello, my name is Andrea Canto, and I work as a technical application specialist at the Flow Panel Design team at Thermo Fisher Scientific Tech Support. Today, I'm going to talk about compensation, what it is and why it is important. Here, you can see in this slide a sample that has been stained with 3 different dyes, Alexa Fluor 488, PE, and PerCP Sci 5.5. You can see the emission spectra of these dyes in this slide, and also you can see the bandpass filters that are available in this particular instrument. The bandpass filters have been represented by the shaded areas. Here you can see that there is some spectral overlap between Alexa Fluor 488 and PE. Therefore, we say that there is a spillover of Alexa Fluor 488 into the PE channel. This spillover here requires compensation to subtract any false positives from the Alexa Fluor 488 signal in the PE channel, as compensation is a process for which we correct the spillover. As for PRCP Sci 5.5, you can see that there is some spillover into the 780/60 bandpass filter, but since we don't have any fluorochrome in this bandpass filter, there is no spectral overlap, and therefore we don't require to apply compensation. So how does this spillover look in our regular plots in flow cytometry? Here you can see a sample that has been stained only with one dye, PE Sci 5.5. On the left-hand side, you can see that there are double positive salts for PE Sci 5.5 and PE Sci 7. However, this sample has not been stained for PE Sci 7. We need to apply compensation to correct this artifact. On the right-hand side, you can see the compensated data. Here, there are cells that are positive only for PE Sci 5.5. The compensation algorithm has worked and corrected the spillover by making equal the median fluorescence intensity of these two populations, the full negative and the population that is negative for PE Sci 7, but positive for PE Sci 5.5. As these two populations are both negative for PE Sci 7, the median fluorescence intensity for PE size 7 in both populations should be the same. Just to finish this presentation, I would like to share with you 6 basic rules of compensation controls to ensure your success in your next flow experiments. The first rule is to have always unstained controls, as your stained sample will never contain an unstained population, as your cells can interact with the antibodies and specifically in different ways. So if you want something that has not been stained, you need an unstained control. This will be essential to assess background autofluorescence. Then the second rule is to have always single stain compensation controls. You will need a control for every dye that has been used in the experiment, and these dyes cannot be substituted for equivalent dyes. So for example, if you are going to use GFP in your experiment, you need to use GFP for your controls. You cannot use FITC as a substitute, and also you cannot use FITC as a substitute of Alexafluor 488. Also, if you are using tandem dyes, the lots must match between the experiment and the compensation controls. The third rule is that the compensation controls have to be bright, at least as bright as the brightest positive signal in your sample. It is always best practice to titrate your antibodies using the cells that are going to be used in the experiment, so then you can use the same amount of antibody to label your compensation bits. If this brightness rule is not met, then your data might be undercompensated. The fourth rule is to have a good separation between the negative and the positive populations in the compensation control, at least one log separation. If the peaks are overlapping, the algorithm for compensation will not be able to calculate correctly the median fluorescence intensities. Then, the fifth rule is to have your control always brightest in the desired channel. So, for example, your PE control has to be always brighter in the PE detector than in any other detectors. Going back to the example in the previous slide, our PE Psi 5.5 control have to be brightest in the PE Psi 5.5 detector and not in the PE Psi 7 detector. If this rule is not met, then the data might be overcompensated. And the sixth rule is to collect always enough events. It is recommended to collect at least 5,000 bits or 5,000 cells for your compensation controls. And if you are using a positive peak representing a rare cell type, at least collecting 100 of these positive rare cell types is recommended.
Hello and welcome to Panel Design Tips and Tricks. I'm Alden, Technical Application Scientist from the Flow Panel Design team. Today I'm going to talk to you about our NovaFluor dyes and how they can be utilized to expand the capabilities of your cytometer. You may have had challenges in designing multicolor panels. And using the current dyes available, there's a lot of spreading leading to poor resolution, which is a result of broad emission and cross-laser excitation. So the NovaFluor dyes have been specifically engineered to address these limitations, having narrow emission and minimal cross-laser excitation. They also provide unique spectral signatures to distinguish them from current fluors. NovaFluor dyes use novel technology. The Phyton technology produces unique fluorescent dyes that are engineered for excitation, emission, brightness, and stability. The NovaFluor dye portfolio currently has 19 NovaFluor dyes, which are available as direct antibody conjugates, conjugation kits. Also, custom conjugation services are available. This is an ever-expanding portfolio, so look for new NovaFluor dyes to come in the future. Each NovaFluor dye includes a cell blocks blocking buffer. This is required for use, which blocks nonspecific binding of novafluor dyes, PE and APC tandems to monocytes and macrophages. When using the blocking buffer, it works best when added to the antibody mixture before adding to your cells. Novafluor dyes are compatible with amine reactive viability dyes, such as our live dead fixable dead cell stains. It is not recommended to use DNA binding dyes such as DAPI, 7AD, PI, or Cytox stains. So you can optimize panels with novafluor dyes. So PE tandems often take up 2 detectors. So in this case, PE Dazzle 594 can be excited by both the blue and yellow lasers. Therefore, it can spill into multiple detectors. This is an example of an 11-color T-cell panel without novafluor dyes. Using Anatune NXT with the 11 colors, you're limited to this type of T-cell analysis, but not further subset analysis. We can update the dyes to add more markers. In this case, we're going to replace the PE Dazzle with 2 novafluor dyes. So this allows us to add 2 additional markers to the panel, CD28 and CXCR4, for further subset analysis. So we can look at naive, central memory, and effector memory T cells from the CD4 and CD8 subsets. Now we have this expanded 13-color panel, which enables greater depth of analysis of the T-cell subsets. So in conclusion, the novafluor dyes are engineered to have narrow excitation and emission profiles to help gain detectors for additional markers. It has less spillover and low spectral spread for high resolution of populations and easier panel building. They're consistent, stable performance with unique spectral signatures. For more information about novafluor dyes and our panel design services, please visit thermofisher.com.
Hello, my name is Andrea Canto, and I work as a technical application specialist at the flow cytometry panel design team in Thermo Fisher Scientific's tech support. Today, I'm bringing you some tips and tricks when choosing your buffers for your multicolor flow cytometry experiments. First of all, if you are using more than one brilliant violet, more than one brilliant ultraviolet, or more than one super bright dye, or one of the 3 of each, for example, or 2 of them, it is necessary to use a buffer to block any unwanted interactions between these polymer dyes. This buffer can be, for example, the super bright complete staining buffer or the brilliant stain buffer. In this example, you can see that in the right-hand side, the super bright complete staining buffer has been used to avoid any interactions between the polymer dyes. And you can see that there is a nice separation between the CD8 positive cells, the CD4 positive cells, and the double negatives. However, if we analyze the figure in the left-hand side, in which no buffer has been added to block the unwanted interactions between the polymers, you can see that there is an artifactual population appearing. If you are using a novafluor dye in your panel, for best results, it is necessary to use the cell blocks buffer. This buffer has to be added to the antibody cocktail before adding the antibodies to the cell sample. If you are planning to stain any proteins that are located in the nucleus, it is important to use a buffer set for intracellular staining that has been formulated specifically for nuclear proteins. For example, you can use the FOXP3 transcription factor staining buffer set that has been specifically formulated for intranuclear stainings. Another consideration is that when we are staining for phosphorylated proteins, this usually requires methanol permeabilization. But it is very important to note that methanol destroys PE, APC, PRCP, and their tandems. Therefore, it is not possible to add methanol to cells that have been already stained with antibodies that are conjugated to PE, APC, PRCP, or any of their tandems. Instead, it is best to save these fluorochromos for staining cells after washing away all the methanol. Last but not least, if you are planning to perform surface staining after fixation, it is important to check if the antibodies is still recognizing the epitope. This is because most of the flow cytometry antibodies targeting surface proteins have been formulated for cells that are alive and not fixed. Therefore, it is important to always check if the clone will still recognize the epitope if we are fixing the cell prior to the surface staining. This is something that can be checked in our online resources at Thermo Fisher Scientific website, checking for antibody fixation considerations.
All right. Thank you again, Dr. Haus, for your time today, especially as it is so early for you, and to your team who are providing this valuable service and information. We would also like to thank Labroots for helping us bring this educational webcast to all of you. My apologies, I got my notes mixed up. If you have any questions that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is your service limited to designing panels of a certain size or for specific flow cytometers?
Thank you, Phil. That's a great question, right? As we all are scientists working with different sets of instruments, it's not always true that you only have Thermo Fisher branded instruments. So we are supporting all customers independent from their instrumentation, as long as they provide enough information about their configuration. And on top of that, we are also independent from the size. So if you're coming with a 6-color panel and you want to add one additional target on your list, or if you're working with a 30-color or even up to 40-color spectral panel, we will help you with that.
Okay, great. Thank you. We have another question here. What is the best way to get in contact with the team if we want to use the service?
Yeah, another good question here. So the easiest one is to contact us via email. I highlighted that throughout the presentation. We have a dedicated email address, which is flowpaneldesign@thermofisher.com. And you can contact us in English, but we also offer regional services. You saw my colleagues Saki Young and Takanori joining today's meeting, and they are providing support in their local languages, Japanese and Korean language.
Great, thank you. Another question that we have is, how long does it usually take to get a panel back?
Yeah, you will get for sure a first reply within 24 hours, but after that, it all depends on the complexity of the panel. you can imagine if you have a panel which is 40 colors, or you want that it includes 40 colors, that is a little bit more time consuming compared to a 6-color panel. But sometimes even a 14-color panel can become very tricky if you're going to the limitations of your instrument. So what we are usually saying is within 48 hours, the easy panels, within 5 days, the most complicated panels. But let's say we have bank holiday season or anything like that, we would always let you know how long it will take until we are finished. And in case we foresee a little bit of delay, we will ask you openly if this is still in your time frame, and you can always tell us if this is not the case, and then we will prioritize.
Right, thank you. We have another one here. What is the best approach to try and fix compensation errors?
Yeah, that is a very good question, and we see that a lot with our customers. It's, to be honest, flow cytometry can be a little bit more complicated, right? And you have to be an expert here to ensure that all of your data is something you can or you want to publish. So we have a couple of products which will help you with the compensation strategy, and my team offers, after the panel design service, also help with establishing your protocol, which includes compensation. So my best approach would be here, if you want to know more about compensation and how to put in a strategy, that you would contact my team, and they will discuss that in a lot more detail than I could do that today. And they will set up with you the strategy. They will provide a list of products if this is required. They will also consider what you have already in your lab, and then they will take it from there to explain the optimal strategy for that.
Great. Thank you for that answer. Another question that's come through is, when you design a panel, do you make recommendations for which controls should be used?
Yes, as I mentioned already, very, very true. I mentioned it already, but let me do it again. We are full service. So from the beginning, when you contact us about your experiments until the point where you're getting really good results. So the idea is that we will help you from the beginning till the end. So in case you're having questions about compensation, and trust me, we all have them, then what we would offer is help finding the right products. We offer products for compensation purposes, but we would also help you setting up the protocol to optimize your compensation strategy and so on.
Yeah, thank you for that, Patricia. I think selecting the right controls for an experiment can be one of those tricky things that a lot of people require a little bit of guidance on. So it's great to know that the team provide that. We have a few more here. Can you help point me in the direction of suitable protocols for my assay?
Yeah, so we have a summary already on our webpage. So if you want to go to thermofisher.com and have a look into our flow section, that is a good start always. But as narrowing it down, it can be tricky. Again, saying it's best to contact us and we will guide you to find the right protocols. But we will put also a focus on your research, right? Because it's not one protocol which will work for all sample types. It might be a little bit more tricky if you're going into the direction of spectral flow. But anyway, there are protocols available, and we will help you to make them more suitable for your approach. Phil, I see that we have a lot of questions still in the chat, but we are running out of time here already, right?
Yeah, I think Yeah, we are. So we will answer any questions that were put into the chat and sent through by the customers that we haven't answered already. We'll actually get back to you via email, but we are at time. Thank you, Dr. Haus, again, and to your team for putting this excellent presentation together today. I think it's a really exciting service that you're providing, and it's clear from the audience that have attended today that this is really an area of interest and something that our customers will find very helpful for their science going forward. Again, I wanted to also thank Labroots for helping us bring this educational webcast to our audience today. And of course, we want to thank the audience themselves. Thank you all for those who have tuned in today live and for your interesting questions. As I said, we'll get back to you very quickly, but we are at time today, but we will answer your questions via email, and we can even, set up a meeting or whatever if you want to discuss further with someone in the team. This webcast will be available to view on demand from tomorrow, and Labroots will also send you an email notifying you of when the replay is available. We encourage you to share that email with your colleagues who may have missed today's live event. But until next time, take care, everybody, and goodbye.
Thank you, everyone. Please feel free to put in more questions if you want, or contact us directly.